First, I would like to welcome you all and thank everyone for coming. It's a great honor for me to introduce you to today's eminent speaker for the 14th session, Dr. Sofan Fafal. Dr. Sofan is a PhD holder from IIT Bombay 2012, a senior research scientist worked at Research Center 2013 to 16, and a research scientist at Glenmark Pharmaceuticals Research Center 2016 to 20, a senior manager at Far Pharmaceuticals Research Center 2020. He has many laurels to his name, which include Reviewer for Royal Society for Chemistry Journal (RSC), keynote speaker at Industry Academia Meet, CDRI NIPER Raebareli 2018, Business Excellence Award at Glenmark Pharmaceuticals 2017, BSc International Award 2012, Academic Unit Representative, Academic Affairs, Chemical Engineering Department IIT Bombay 2010 to 12, Best Poster Presentation Award in Research Scholars Symposium Research Showcase to Industries IIT Bombay 2010, research work recognized in top 14 entries and bagged Bioencapsulation Research Grant BRG in Netherlands 2009, Avishka 2007 Inter-University Research Competition and bagged second prize Nagpur, All India rank 99 in GATE 2005. His research and development areas include microfluidic devices or systems development, applications in pharmaceutical or biomedical industries, complex injectable formulation development, liposomes, microspheres, nanocrystals, ophthalmic suspensions, carbohydrate complexes, protein, and peptide. We welcome you, sir. Over to you, sir.
Yeah, thanks. Thanks, Arundhati, for nice introduction. And yeah, definitely, as I mentioned, this will be some new for you people because I'm having background, mixed background, like pharmaceutical and chemical engineering. So I will try to cope up with engineers because my background is in pharma and then PhD in chemical engineering IIT Bombay. Okay. So I prepared my slides so that I mean I can easily tell you or explain you. So what exactly research we are doing at our pharma industries. Just be patient, and I will try to make it as interesting as possible from my end. Whenever you want, you just ask questions. Stop me. Don't hesitate and ask questions for clarity. So I'm just presenting my slides now. I think share option is not there. Can you give me the privilege for sharing, Arundhati? Who is? Hello.
Yes, sir.
Yes.
Yeah, actually, that I'm not able to click on share option.
Sir, there's an arrow down there. If you click, you may find it. Because I guess
Yeah,
it's.
I'm clicking it, but it is not highlighted. I'm not getting able. I'm not able to click on that share option.
Uh,
So I think one need to assign the privileges. Just I will try. No. Sopan. So no.
Wait, wait.
Yeah.
అక్కడ వరకు వెళ్ళి వచ్చిందా ఓకే థ్యాంక్ యూ వెరీ మచ్ థ్యాంక్ యూ కలర్స్ వైట్ ఇక అయిపోయింది మందాలు సెట్ ఇది మళ్ళీ ఆకులు వైట్ పెట్ట డైరెక్ట్ ఇట్లా ఫ్లవర్ పెట్టరాదా అట్లాగనే కొన్ని ఇవ్వండి అరగాయ అంటారా ఇంకోటి మీద ఇంకోటి పెట్టాక
You can try. Simba,
Sir, can you click on the share option? Present now?
Yeah, share option that
Yeah,
is not
present.
actually enabled for me.
1 minute.
Yeah, moderator needs to, I think, enable it for me. Meanwhile, I'll just share my slides with Professor Jairam. If it is not possible, then
Yes.
you can present.
you will try.
Right. I just shared with you.
So you can share your screen.
Just a minute. No, again, can you see here actually that shared option is not enabled. Just a minute. So Professor Jairam, just check your email. I just shared my slides. So from your end, you can present it.
Okay.
I'll just send. Yeah.
Is there any technical problem in sharing, sir?
Yes, yes. I'm facing this. The sharing option is not enabled for me. So what we'll do, I shared my presentation to Professor Jairam, so he can present then.
Okay, okay.
Yeah, actually somebody made me co-host, but still.
You are pressing that arrow button, na? Up arrow button.
Yes, yes, I'm pressing it, but that share button is not enabled. Like,
Oh,
right.
maybe then, sir, you can log out and log in again. Something if it works, then you can try
Mm.
like that.
Wait.
Or better, I mean, Professor Jairam can share the slides.
Okay,
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 okay. Wait.
okay. No problem, sir.
Right. Yeah, these are glitches, online glitches.
Yes, sir.
It's better actually physically we come there and present. But yeah, still,
Yeah. Hopefully next year we can have the similar
yeah,
program in a face-to-face mode.
yeah, good.
Okay.
Okay, thank you. Okay, just a minute. I need to set up. Presentation mode is you are already there, right? Okay. Yeah, I'm starting presentation. So, thank you uh, for giving me this opportunity. And today, as a part of drug engineering, ATAL or AICT program, so I'm going to present complex injectable formulation development. I have around 10 years experience industry and total of 16 years research plus industry experience. I mean, academia plus industry experience. So, okay, you can move to next slide. Yeah, next slide, please. Hello, sir. Yeah, thanks.
Yes, sir.
Okay. Yeah.
Audible. Yes.
So this, yeah, this outline, like introduction to complex injectable. Then actually why we are choosing complex injectables. Then challenges, particularly in development. Or here we can discuss about drug engineering. The challenges are basically three: regulatory, patent related, and formulation and characterization. So formulation is actually engineering related challenges. So I will be focusing more on that. Then of course, pharma industry, everything is there. I mean, this is called FDA, Food and Drug Administration. So they have laid down very well defined paths for development, and in general, pharma industry follows their well laid down regulations. I will just explain how this FDA is encouraging industry, academia collaboration and all, and then I will just um, for example, for just better understanding, I will give one example of complex injectable, that is liposomal drug delivery system. So this is actually currently being used for COVID vaccine by Pfizer also. And last one is conclusion. So next slide, please. Yeah. So this is just overview of Indian pharma industry. So as you are knowing, India is called as world hub for pharmacy or medicines. So in that, if you compare with US, so 51% share is there from Indian pharma generic companies for US healthcare, and worldwide it is more again, like rest of the world, South Africa and all. So so nowadays. There are several off-patent products are there. In general, just for your information, generic means what we are doing in India. So there are innovator products. Though few of our in companies are also providing or developing innovative product also. But more or less, trend is that US company will provide the reference product or innovator product after their patent is off. Indian companies are following and preparing that copy of that drug that is called generic. And in that, so whatever the complex products are there, though patent is off since last 10 years, it's very difficult to copy it or to manufacture similar product. So what are the challenges and all? This is the presentation today I am going to present. Yeah, next slide, please. I hope I am audible clearly. Okay.
Yes,
Yes,
sir.
sir. Yes, sir.
Yeah, thanks. So, what are complex injectables? As of now, all of us have come across injections. Mostly, they are plain solutions, colorless water kind of injections, or without particles or anything like that. So, these are plain injectables, or simple injectables, I can say, or conventional injectables. And what are the complex injectables? So, basically, complex injectables are designed mostly most of the time drug engineering is involved, and they are designed to have the better compliance. I mean, with respect to product quality attributes, or with respect to product safety, or easiness to the patient, or we can call it as patient compliance. So most of the time, I mean, you can see these complex dosage forms are basically divided into multiple subtypes, like complex strength, complex route of administration, even quality. As I already mentioned, simple generic, you are aware about what I am talking. This is plain solution or simple solution, but complex generics are they are either containing complex active or whatever we call it as drug in pharmaceutical industry. So drug itself is a complex. For example, proteins are like insulin. It's very complex to produce. Then complex formulation. So mostly here drug engineering part is involved. Complex formulation. So these are having microparticles, nanoparticles, drug crystals, so like that. And most of the industries are focusing on these complex formulations or where the drug engineering is involved. And other parts like complex route of delivery, like suppose you want to deliver it in your local part or something like kidney. So that is complex, but it's something more complex to the physician, not for engineers or developers. And of course, last one is complex drug device combination. So here again, engineering is involved, and these are in fact a delivery device is complicated here. For example, there are some nasal DIP inhalers or sprayers like that, no? So every time it should give the accurate dosing and blah blah. These are the complexity with respect to the device. But as a part of this product program, I will be focusing mostly the complex formulation or drug engineering. Next slide, please. Okay, just for example, I am giving here the type of complex injectables or generics. So as I mentioned, complex active ingredients. Most of the time, peptides, proteins like insulin, mixtures, some natural source products are also considered as complex because reproducibility is a problem. Then complex formulation, which are, as I mentioned, these are liposomes, microparticles, nanoparticles, these iron colloid particles. So these are used for iron deficiency or anemia, most of the time in pregnancies. And it's very difficult. Your hemoglobin levels become very low, and if you are on tablets, everyday tablets, you for 1 month again, still you are having deficient iron deficiency. So there are certain formulations you give once, and for 3 months your iron level will be maintained. So that's why there is a complications, not complications, complexity in that active, and we are conjugating it with the some carbohydrates, and also complex route of administration, locally acting, as I mentioned, kidney and all things. And last one is the drug device combinations. Next slide, please. Okay, and why companies are focusing for this? So as I mentioned, there is me too generic. Lot many players are there for plain injectable solutions. In simple solutions, nothing is complicated. Just mix, dissolve the active ingredient, and it is ready for your injection. Just you have to maintain the sterility and all, of course. And of course, a price is very high for complex things because development involves lot many controls and timelines. Of course, as I mentioned, there is a great patient compliance. Nowadays, a lot of our companies are preparing more suitable advanced drug deliveries for India market and of course world market also. And also to neutralize like Me Too space, to have the more margin over the other companies. There are fewer competitors for these complex injectables. And of course, nowadays most of the companies, including foreign or MNCs, so they are focusing mostly now these complex injectable drug products, and comparatively like oral solid drugs like tablet, capsule and all, their share is declining because nowadays biologics drugs are coming in picture. So though we are behind some 10 or 1 decade than other Western countries. So now at India, we have 10% biologics share and 90% NC, or we can call it as chemical entities. But now future is changing. So next decade will be for biologics. So biologics means, for example, insulin is a biologic product. So we cannot manufacture it in lab. So we have to. I mean, we can do it, but it is not chemical reaction. It is you can say engineering, or I can say cell biology basic product. Next slide, please. Okay. Yes. Just for example, for your better understanding, there is one molecule called as doxorubicin hydrochloride. So this is used for cancer actually. And here, for your information, I am just explaining conventional versus complex. So these are 100 nanometer liposomal lipid balls containing the drug, encapsulated drug inside that lipid ball, you can say, and size is 100 nanometer. So because they are actually encapsulated in nanoballs, so toxicity is very negligible compared to conventional one. Because conventional, you cannot inject more than 10 mg of drug. So there is large, I mean, chances are there you will get a cardiac toxicity. So like that, since it is encapsulated, the toxicity is removed now. And second parameter is called T half. I mean, once you inject at means how much time your injection is giving effect. So just for layman's language, I'm saying here. So for conventional, it is 20 minutes only, but for this, it is 50 hours complex formulation. And of course, site specificity, I mean, or targeting. So you are aware, we are saying treating cancer is very difficult because cancer cells are our own cell, and your drug molecule don't or medicine don't recognize whether it is cancer cell or it is normal cell. That's why we are saying that if you are injecting or treating the cancer patient, he's facing a lot of problems, health related problems. All hairs will be going off, and he's feeling a lot many heats and toxicity. So these nanoballs, particularly, they are targeting only the cancer tissue. So that is the major advantage. I am saying like compliance to the patients, and that is reflected in cost also. You can see here, the cost for conventional is $5 to $10 only, but for this complex liposomal formulation, it is above $1,200. Just for your reference, I'm highlighting here. Similar thing is with amphotericin. It is same formulation again, liposome, and below is the nanocrystal as I'm talking. So compliance point of view, we can see here. So duration for conventional is 1 day tablet, and for this complex injectable, it is 90 day. So this is used for psychiatric patients, and it is not possible for psychiatric patients. Yeah. Do- Does have somebody have question?
ఇది
No,
కూడా ఒక
sir. No.
వర్క్ అయిపోయింది. అక్కడ
Okay.
ఉన్నాయి ఇవి ఇక్కడ.
Some disturbance is there. Please wait, wait.
Okay. Yeah. So as I mentioned, that drug is released for over 3 months with this complex injectable. And same case is with triamcinolone. This is
తీసుకుంటారా
knee
వ్యాక్సిన్?
injection.
తిప్పుతుంది
Yeah.
తిప్పుతుంది అంటే నాకైతే తిప్పింది.
Sir, please wait.
ఇంకా రాలేదట కూడా. ఇప్పుడు వచ్చిందా? ఏమో పొద్దున అప్పుడు రాలేదన్నారు.
Moderator, you can mute all except me.
Yes, sir. Just a minute.
ఇవాళ ఇవాళ వినరు వినరు ఇవాళ వదిలేయాలి. ఏం కాదు, ఇవాళ సెలబ్రేషన్సే కదా.
Please wait. Just a minute, please.
Yeah. You can move to next slide.
వాళ్ళే తెచ్చారు. వాళ్ళు వేరుస్తలేరు. ఇప్పుడు వెమ్మంటే తెచ్చారు. వాళ్ళు వేరుస్తలేమన్నారు. తెలియదు మేడం అన్నయ్య వేరుస్తారా అంటే వేరుస్తలేరు కానీ వినరు. కాన్సన్ట్రేట్ చేయరు. డ్యూటీ చేస్తాను.
Yeah, Professor. I think there is an option on screen. You can mute others. Central button. Yeah, you can mute others. On screen, presentation screen, it was showing me on center. If you are moving cursor, it is showing mute all.
Sir, we don't have that.
Okay.
Please wait. I'm
Yeah.
calling for your administrator. Please wait.
Sure.
వాళ్ళు కూడా ఉన్నారు. వాళ్ళు ఇన్విజిలేటర్స్ ఉంటారు కదా వాళ్ళ అప్పటికప్పుడే డిసైడ్ అవుతుంది. అప్పటికప్పుడే అడిగిన అటు ఇటు అటు ఇటు ఫోన్లు అయిపోయింది కదా.
Yeah, thanks. Next slide, please. Okay, few of the slides I will just faster. This is just overview. We can move ahead. How companies are focusing now in complex things. Next slide, please. Yeah, next slide. You can go. Okay. Yes, this is just a chronological order. As I mentioned, so these are traditional generics, low complexity is there. This is R&D complexity I'm talking about. Modified release dosage forms, injectables, plain injectable I'm talking, simple, transdermal, dermatological, complex injectable, respiratory because this is containing drug device combination. Biosimilars, what I am talking about is biologics. So these are the level of complexity. Next slide, please. Yep. So these are the challenges. Just for explaining better. So legal for conventional, there are no legal challenges, but for complex, there are a lot of many. Like citizen petitions, patents are there. So it's tricky. Uh, Second one is development timelines. So for conventional, it is less than 1 year we can develop the product, but for this complex, it takes around 4 to 10 years for getting approvals or for readiness, final readiness of your product, development to production level. Of course, huge part is, I mean, huge budget is required for clinical studies because this is complex thing and a lot many nanoparticles and microparticles. So now you are modifying your drug molecule, and modifications not only give you positive effect but sometimes give a negative effect also. That's why clinical studies are required for complex generics. And of course, cost of development is huge for complex, definitely. A regulatory pathway, as I mentioned, it is well defined for simple, plain, or injectables, but not defined for complex generics. Of course, this is the main bottleneck of characterization. For conventional, it's easy, and for complex, it's very difficult. And manufacturing controls, like for conventional, it's very easy, simple. You can manufacture within 1 or 2 vessels. But for these complex things, we need to develop automations and lot many in process tools are required. Lot many engineering controls are required, and almost skills are required. So which will again invite for huge investment. Next slide, please. Yeah, these are just few of the examples. Like I am saying, long-acting parental injectables. Long-acting means 1 month, 3 months, 6-month release also are there. So this is from patient compliance. It's very good. I mean, having the long-acting injectables. Definitely, this is case-to-case dependent. Like as I mentioned, for contraceptions, 6-month injections are there. And what they mostly the engineering part of that drug release, long-acting release is that they are having microparticles, or these are polymeric particles where drug is actually embedded in polymeric capsules, like implants and inserts are there. Then like liposomes, what I'm saying, what I talk is these nanoballs. Suspensions are there injectable. Then again, these iron and carbohydrate complexes are there, and nanocrystals are there. And of course, for ophthalmic emulsion suspension are there. There is new category again in US FDA or Western countries ask nowadays. It is abuse deterrent formulations. So these people are having issue with teenagers. They are actually using lot many abusive drugs. They are purchasing from pharmacies and using it for their actually doing the misuse. So to avoid that, US government is investing almost 50% of their health insurance or health budgets for preparing abuse deterrent formulations. They are again new category of complex formulations. Next slide. Okay, manufacturing challenges. Just for your information, basically we have Q1, Q2, Q3. We call it as quality to equivalence, quantity to equivalence, and Q3 is this is called a physicochemical property equivalence. Basically, if you are preparing generic drugs, and just one example I'm putting here for your information, for your better clarity. Like for plain simple solutions, manufacturing vessels are only 2 to 3 are required. For I am giving an example of liposomal formulation. So here 17 manufacturing vessels with skids and automations, and of course a lot many in-process tools are required. Manufacturing steps, it's 1 or 2 step only, but here a lot many steps are involved, around 5 to 7 steps are there. Specialized equipments are required for these liposomes. There are 4 to 5 equipments, and of course, batch processing time. So yeah, it is 1 to 2 hours takes for simple solution, but 5 days are required for these nanoballs, or like we can call it as liposomes. Next slide, please. And second thing is again here, as I mentioned, microparticles. These are actually having complex inactive ingredients. I mean, here the polymer, which is used for to control the release or to extend the release, is itself complex. And the here major problem what we are facing at R&D, because everybody is knowing at research and research development we are taking batches in liters only. I mean some 2 or 5 liter batch only, but for production requirement is for 100 liter or 500 liter, like that. And what happens that now the scale is changing, your lab scale is changing. I mean for manufacturing of 1 liter batch it will take say 5 hours, but for manufacturing of 100 liter batch it may take 50 hours. So during that what happens, so this polymer get degraded, and whatever the quality of your product manufactured at R&D, it is not going to meet when you are producing at large scale. So here actually your contribution or our engineers contribution is needed. We need to develop devices or manufacturing setups such that um, so that scale dependency can be neglected. And that what I'm talking exactly at the last of my presentation I will present that. So next slide. Okay, again same thing. When you are changing manufacturing methods, you will get the change in your parameters. So there are 4 different manufacturing methods are used, and every time you are getting different profiles, release profiles. What I am talking. So that is again one major challenge from engineering perspective. Yeah, that can be controlled again definitely. So that part I will cover at last. Next slide. Yep. And of course, as I mentioned, there are certain like regulatory related challenges also there, because there is no clear-cut defined path, and everything, lot many sciences involved. So it is basically for complex development is basically collaborative research, I can say. So here, lot many industry academia collaborations are expected, because industry is mostly driven by time and money, of course, and that's why they don't get much time for having core research. But academia people have means like PhDs and masters people, they can take these challenges, they can develop, because academia is more free. I mean free thinkers are there, they don't have time bindings like what we are having at industry. And that's why regulatory agencies are also encouraging by providing the grants for academia also. So you will get that all details from USFD website. Lot many grants are available. Crores of grants are available. Just we have to apply and get that grant. So that yeah, somewhere I have mentioned that side. Okay, next slide. Okay. Yeah, this is what I am talking. I was talking. So in like example in 2016, around sorry, this is something guidances are there, but yeah, 15 grants are actually provided by USFDA. And for your information, one grant contains at least 5 crore funding in Indian rupees. And lot many IITs, institute like IITs and NIPERS and some government colleges or engineering colleges are also applying for these grants, just to resolve issues related to the manufacturing. For example, engineering colleges are asking for a grant, and drug development pharmaceutical colleges are asking for that grants. Next slide, please. Okay. Second major challenge was basically characterization. So here again, yeah, academia can help us in better way. So academia can develop the scientific based sound methods to discriminate between how drug is going to behave inside the body. Basically, characterization helps you to determine the drug's behavior outside the body and how it is going to mimic inside the body. Like example, I showed in last slide. There was a difference in release. Release means what we are doing outside of in the beaker. We can call it as dissolution testing. Basically, we are putting our drug particles or formulation particles, and with certain conditions, temperature, and RPM, whatever. So that drug amount of drug coming out of that carrier, per I mean the time dependence is there. So that is actually getting mimicked when you are injecting inside the body. So that's why pictorially I am mentioning here. Lot many testing outside means lab testing apparatuses are there, like HPLC, GC, and all, and they are mimicking drug performance, how it is behaving, how is it is going to behave in body. So there should be some correlation, good sound correlation. And here in foreign countries, lot many this part, whatever ex vivo or in vitro testing part is done by academia, and then ready made they are providing to the industry, or they are having collaborations. Next slide, please. This is something regulatory. We can skip. We can skip. Next slide, please. Yeah, again, next slide. Here again, I have already mentioned we need help from academia. Next slide, please. Okay. The just short conclusion of whatever I am talking here for challenges in developing complex injectable dosage forms. So, of course, there is increasingly crowded traditional drug markets. To counteract that, we need to focus on complex generics, like and also to enhance the compliance, patient compliance. We need to focus on complex generics, and of course there are certain drawbacks, like development cost is time and cost is very high, and as their name implies, it is generally difficult to establish equivalency, as I have mentioned, between reference product and complex copy of that product. And this bottom line of this is like a substantial commitment and resources is required. Of course, there need to be good collaborative research between industry and academia. And of course, however, if we are doing successfully, it rewards are very high. For just I have for comparison, I have mentioned here $5 versus $1,200 is the cost of 1 unit of vial. Next slide. Yeah, I'm just giving for better understanding one example of complex product. So many times I have mentioned that nanoballs or liposomes. So these are nothing but pictorially you can see here. So basically they are made up of one basic chemical entity called it as phospholipids. So for presentation we can see a blue color is head of that phospholipid, and that tail is carbohydrate chain. And I'm just explaining here, top layer is actually called it as bilayer. When it comes in contact with water, this blue head is hydrophilic or water loving, and that tail, carbon chain tail is actually hydrophobic, water hating. Just to avoid interactions with water, they are aligning themselves, head facing outside and tail inside, so that water will be now inside and outside, of course, and it forms structure like bottom, what I mentioned here. Pink color is head now, and to avoid the thermodynamic or free energy issues or thermodynamic stability, it gets encapsulated or it gets enclosed into nanoball, and these are called as liposomes. Nowadays, these liposomes are used by Pfizer for COVID vaccine. So inside the center core, they are encapsulating a drug or call it as mRNA molecules, basically, and they are acting just to means they are a major ingredient for this COVID vaccine. And carrier is liposome. Just if you are, yeah, we have sufficient time. Sir, can you please click on this link I have given here, or if it is not clickable at your end, copy it and paste it in your browser. So it is very good video,
1 minute.
3-4 minutes. Video. Yeah. I know many people might be not having relevant background, so I'm trying to explain it. Please prepare and unmute it. What's happening? Sorry, what's happening? Yeah. Oh, oh. Sorry. Huh? It's running at my place.
1 minute.
Yeah. Yeah. You can basic. You can. Okay. Go back. There is. Yeah. First video 6.23 minutes is there. No. Yeah. Click that. Yeah. Play it with sound.
Is commonly removed by rotary evaporation, resulting in a thin film of dry lipid. Some solvents can be removed by lyophilization, resulting in a dried lipid cake. An aqueous solution containing water-soluble compounds to be encapsulated is then added to the lipid film or cake to hydrate the lipid and form the liposomes. The aqueous suspension must be heated above the phase transition of the lipids in order for the liposomes to form. The resulting multilamellar liposomes consist of many concentric lipid bilayers. While all or most of a lipid-soluble compound will be incorporated into the bilayers of these liposomes, the amount of water-soluble compound entrapped is usually very low because these MLVs have a small captured volume. Very little aqueous solute penetrates the onion-like layers of hydrated bilayer lipid. The captured volume of these MLVs can be increased by freezing and thawing the liposomes several times. Repeated freezing and thawing ruptures and reforms the MLVs, resulting in an increased number of liposomes with fewer layers of lipid and more aqueous space inside each liposome. Morphology. The aqueous solute is now uniformly distributed inside each liposome. The thin film method is limited to laboratory scale, while the reverse phase evaporation method can be used to produce milliliters up to thousands of liters of liposomes. Lipid is dissolved in organic solvent along with any lipid-soluble components. The aqueous compound is added to the lipid solution. This biphasic mixture is then emulsified by some form of high shear mixing. The organic solvent is evaporated from this emulsion, forcing the lipid into the aqueous phase, where it organizes into bilayers and forms liposomes. Additional aqueous phase is added in order to complete the liposome preparation. Again, the temperature of the aqueous phase must be above the phase transition temperature of the lipids in order for liposomes to form. Unlike MLVs produced from a thin film, liposomes produced by reversed phase evaporation usually have one up to a few bilayers per liposome. Like freeze-thawed liposomes, these liposomes can be multivesicular and have uniform aqueous solute distribution. Liposome characteristics can be optimized for a particular formulation by systematically varying the process parameters. Many variations of these methods are described in the scientific literature. Liposomes produced by these methods have a
Like
broad
I mentioned, there are multiple steps are involved.
which can
So this is
produce
one step for sizing.
total
Yeah.
distribution of liposomes in the 100 nanometer range is high pressure extrusion through membrane type filters of a fixed bore size. The stainless steel device, which supports the filters used for extrusion, withstands internal pressures in excess of 1000 psi. Extrusion cannot be accomplished with standard liquid filtration devices. Larger extruders are pressurized by nitrogen, while extruders handling volumes less than 1 milliliter utilize gas-tight syringes to generate pressure manually. Extrusion must be carried out above the phase transition of the lipids in the liposomes. In order to achieve a narrow size distribution, the liposomes must be passed through the filters 10 times or more. The liposome suspension becomes more translucent during extrusion, indicating that the liposomes are becoming smaller. The lower size limit of a liposome is 30 to 60 nanometers, depending on the type of lipid used. High shear methods are used to reduce multilamellar liposomes to unilamellar liposomes at their lowest size limit. Most water-soluble compounds must be present at the time the liposomes form to be encapsulated. Some ionizable compounds can cross the membrane in their neutral form.
This is doxorubicin molecule, what I am talking.
Salt form inside the liposome. This remote loading technique allows drugs to be loaded
You can
into
see
liposomes,
how smartly they are engineered now.
but is not widely applicable. Some ionizable compounds can also be loaded into pre-made liposomes with a pH gradient. In this example, the neutral form, but not the charged form of a compound, can cross the membrane into the liposome, but can't escape due to the high concentration of protons inside the liposome. Lipids organize into bilayers due to the presence of water. When the water is removed by lyophilization, the bilayer loses its structural integrity. After rehydration, the liposomes have a different size distribution and have leaked their entrapped contents. Some aqueous solutes, such as sucrose,
Okay, you can stop it now.
water at the lipid head group.
Yeah, go to our slides. So just wanted to say these are actually engineered very nicely, and there is a lot of science involved in there, um, designing these drug deliveries. And as I mentioned, there are multiple steps are involved here. So sizing, first of all, formation of bilayers, then encapsulation of drug, sizing, then again lyophilization and all these things. Purification is also there. So go to the next step. So as I mentioned, this takes around 1 week for manufacturing a single batch. So here we as engineer can help industry, pharma industry, to reduce that time and to avoid lot many that unit operations. So next slide. Yeah, next slide. Go. I have already mentioned there hydrophilic and hydrophobic. Yeah, sorry. Yeah. Yeah, these are just advantages of these nanoballs called liposomes over conventional things. So it is these are site specific, as I mentioned. They are particularly specifically targeting the cancer tissues. Then of course it gives extended release. As I have showed, it gives up to 72 hours release, or delayed release also, we can call it as. These are helping for patient to avoid the toxicity and to get better compliance. Instead of taking injection every day or tablet every day, we can delay it for months or like a few weeks also. Yeah, we can skip few of the slides. Just go to next. These are just characterization point of view which are not relevant. Yeah, as I mentioned, these are currently in industry. People are using multiple step things that are shown in the video. Next step, next slide. Yeah, just we can skip few of these slides actually. These are very basic things with respect to, of course, how to design these drug molecules. Uh, Again, just I will just try to spend few moments here. Yeah, so all right, skip. Yeah, sometimes we have to design these hydrophilic polymeric chain on the surface of these liposomes because our physiology or our body expels whatever is foreign material, and to fool the body we have to attach this kind of polymers, hydrophilic polymers. We call it as stealth liposome. Stealth means hiding, or we are hiding that inside this polymeric brushes, and body now doesn't recognize, yeah, it's foreign body or foreign particle, and then efficacy or effectiveness is then enhanced. Next slide. Hmm. Yeah, these are just some of the properties. There are certain 25 to 30 different characters and techniques are there. We are not going through this now. Just next slide. I just wanted to, focus on how we as an engineer can help to the pharma industry. We can go to the we can skip few more of these slides. Yeah, go ahead. Go next. These are very basic chemistry or characters and related things. These liposome, actual liposome, yeah, sir, just go for previous slide. I will just want to show here. Whatever we have seen, these are theoretical images and videos. Just previous slide. One more previous slide. Yeah, this one. These are actual images. This is called transmission electron microscopic images. You can see that scale bar is 200 nanometer. So this is 200 nanometer to or below 200 nanometer. Single nano balls are there, and these are dummy nano balls. And this big image at right hand side is having this doxorubicin, this coffee shape bean. No, so centrally you can see the rod shaped thing is there. This is nothing but monomers of your drug molecule got entrapped inside these nanoballs. And you can see these are now protecting, these drug molecules are not free to expose other body parts other than cancer tissue. Okay, and they are protecting there, and selectively they are going inside the cancer, detecting the cancer tissue or that tumor, and releasing their material or load inside that tumor. And now there is lot many advantages are there. The toxicity related things are negligible. And that bottom left picture is actually having multi-laminars. In both of these are single layer. You can see here multi-layers are also there. So multi-layer particles are used for different applications, not for cancer one. Just for your curiosity, how these look like. These are all 100 nanometer size particles. You can see here. Actually, they are looking like this. Yeah. Next step. Next slides. Yeah. Next slide. We can skip this. Yeah. Skip this. You can skip this also. These are just actual images. Skip this. Next slide. Next slide, please, sir. Again, next slide. I'm talking actually from more or less these are pharmaceutical related things. Next slide, please. Yeah, next slide. We can go next slide. Okay. Yeah. Yes. Okay. Yeah. From this slide. Just I wanted to focus here. Whatever the background and story I told about developing or engineering these drug particles in pharma industry, at particular R&D, how we as engineer can help for this drug complex drug engineering or development of these complex drug formulations. So, so there are definitely certain different techniques are there. Instead of going for this conventional 5-6 steps, we can see. I just for representation I have mentioned here. Again, these are old methods. One is of course emulsification by application of some sonicators in probe and all. And second is spray drying. Solutions are needed in spray drying, and we can formulate these particles within a single step. But definitely having good control on these devices is very difficult. We can go to the next slide. These are alternatives for whatever conventional we are producing here, manufacturing here. So next slide, please. Yeah, yeah. These are somehow we can say interesting, and some new techniques are there. So the top one I'm talking is actually is electrospinning. We can call it as electrospinning. So here we are having these amplifiers or high volt source, and your drug molecule is yellow. I'm just representing here. It is filled in syringe, and your polymer molecule is now filled in this uh, green color syringe, and it is fixed on some syringe pump so that it can have some control flow rates, and we are applying high voltage to the needle. Okay, and based on your voltage, how much voltage frequency you are modulating, and of course the viscosity and flow rate, so you will be able to produce these nano to microparticles ranges here. Definitely these are very new, novel and single step technique, and this electroformation is somehow in nascent or at R&D level itself, or at academia level, I would say academia level. And second one, our bottom is actually having this microfluidic devices. So microfluidic devices is actually basically nothing but the full flow chemistry. It is more or less, you can say, fluid dynamic related things. So here again, same representation is this. You can pass polymer and you can pass drug fluid simultaneously, and again you can focus it with the aqueous phase or anti-solvent phase. And based on this flow rate, simply we have to modulate the flow rate and concentrations, of course, viscosity and all. And you will be able to produce in a single step control manner particles. Go to the next step, the next slide. So yeah, next slide, please. I'm not able to visualize it yet. Yep, this is one just example I'm talking here. So same thing, these PLGA microparticles are being produced using this microfluidic technique. So you can see your aqueous phase, or we can call it continuous phase. Continuous phase is having nothing but the your aqueous molecule and surfactant, and of course your dispersed break or drug phase is there centrally. So at bottom picture you can see this 1% polyvinyl alcohol is surfactant in buffer or aqueous phase, and your polymer and drug in solvent. Okay, and they are mixing control manner here and forming the droplets, and these droplets you can see actual droplet in that bottle, how they are getting form here. Next a zoom picture of that bottle. Okay, and again it was at academia level only. This technique is called it as microfluidic technique basically. So everything is done at micron level. So fluid behavior at micron level is very different than fluid properties, actually very different than at bulk level, or whatever we are doing in bulk mixing and all, no. Because at bulk there is a lot of turbulence is there, but at micro level or micro channel level, that turbulence, and we have very precise control on mixing and droplet size. So next slide, please. Yeah, you can say, just for your reference, one outcome I am just comparing here. So left hand side is having simple bulk emulsification technique, and in that we have to purify it. I mean, we have to sieve that particles above and below, or we can say we are using chenni. Okay, so if your target is 500 50 micron, 50 micron, you have to sieve it through above particles 60 and below particles say 30 micron. Whatever in between is coming is picture, you can see polydispersed things are there. Then they are again after having some more processing like sieving. And left right hand side is outcome of this microfluidic technology. You can see without any further purification step, single step, you are able to manufacture very uniform particle. And this is the crux, this is the base, how we are going to modulate, or how we are going to control the release of these particles. So huge difference is there. And here definitely this can be done by engineers only, of course, because pharmaceutical people, they are expert in drug properties, and of course they know more about physiology of body, pharmacology, medicinal chemistry, and all this stuff, impurity and all. Next slide, please. Yeah, next slide. Previous slide, please, sir. Yes, this is my research slide actually. This is from my publication. I started in doing this microfluidic technology in 2007 at IIT Bombay Chemical Engineering Department, and this was my paper published in 2013. You can see here a simple. I mean, you can see at middle actually picture. So as I mentioned, I am in mixing that medicine in aqueous phase or water, and these phospholipids in alcohol, and I am actually sending through the syringe pumps, and at the interface it is actually annular now, coaxial, you can see coaxial. Bottom these circles are there, you can see aqueous phase is blue color, and or water is blue, and your lipid and drug is red or pink color. Okay, and they are going to mix at interfaces, and at interface it forms the nanoballs, and this blue color image is actual photograph. Instead of this alcohol or lipid, I use our fountain pen ink just for your representation, pictorial representation, how it looks at our eyes. That blue color, you can see that mixing regime is increasing here at the end of the tube, and these are actually 500 micron channels. Yeah, exactly, these are 500 micron channels. I can reduce up to 40 micron, 40 micron. Yes, and this article I published in 2013. Yeah. And now, as I mentioned, last year Pfizer has adopted this technology for manufacturing the COVID vaccine, basically, and means within one decade, from academia to industrial level, it has been moved now. This is something good. Basically, I didn't patent, but I published this thing. But nowadays in 2020, somebody has patented this technology. Yeah, next slide please. And this is actual outcome, as I mentioned, this is means ESAM, I can say, environmental scanning electron micrographs. So you can see these are actually from my article itself. So output is very uniform, around 150 nanometer particles are there, you can see here. I just compared it with two different techniques. So you can see say around everything is centered in between, say 120 to 200 nanometer basically. And for convention technique, lot many 5 to 6 steps are there. We are not able to produce this kind of uniform single uniform particles. So all these 5 days work needed at our industry, that can be reduced to few hours only, because I am doing single step thing here. And that's why this Pfizer has adapted this thing, because COVID, we need lot many formulation, well, lot many vaccine shots actually, and it was very challenging if we would have proceeded with conventional things, because taking one batch takes around 1 week, but here it takes around few hours only. Next slide please. Yes, this is just from recent article, recent news. Actually, I am, for your representation, I am showing here. These micro devices are looking. These are now parallelized actually. So just for reference, they have mentioned one coin here. This is the size, very small, very compact, and it is able to produce hundred of liters actually, liposomes or nanoballs. This is just for your reference. I am talking, giving some news articles. Yeah, uh, screenshot here. Next slide, please. And yeah, this I have, as mentioned you, this is being used for COVID vaccine. This is again I got from Cash, and there are certain articles also where in Nature, Pfizer have published. You can see whatever I shown in my article. So lipidic phase and aqueous phase, they are being mixed at these micro channels, and output is these nanoballs having vaccine conjugates actually on surface. Okay, and basically, coincidentally, same lipid balls are manufactured by Pfizer also. And at the end, I was attaching polymers. They are attaching this basically peg moiety. I mean, this is actual conjugate for forming antibody or antigen, you can say. Yeah, this surface, COVID surface, no? These are forming, yeah. And as a bottom, you can say this single channel, they are just parallelize it in multiple. The advantage with this technology, as I mentioned, there is scale dependent issue for our pharmaceutical industry with conventional things. So 1 liter to 100 liter, I need to again re-optimize everything at 100-liter plant scale. But here, whatever and at R&D, I can optimize on single channel, and at production level, I have to just parallelize the channels, keeping all parameters same. I have to just integrate that thousands of channels, and of course everything will be fitted on the desktop size only or your CPU size only. That's all actually. No need for bigger vessels, no need for skids, because for just your information, one skid, automatic skid for multiple controls having multiple steps, it takes 500 to 1000 crores for under R&Ds at plant level. And that's why these complex products people are not daring, because of lot many huge investment is there. But we as engineer can help these pharma industries by providing such novel technologies. Because definitely there is lot many industry or lot many academic efforts are there put here for this kind of technologies. And that's why we are expecting from you institutes also, academic institute also, do something like this. And pharma industries are welcome, I mean, they will welcome to procure, welcome to collaborate, or welcome to purchase your technology itself. So yeah, next slide please. Yeah, thank you. Just we are expecting just pictorial representation is there. Academia industry collaboration should be there. If India want to actually compete with these Western countries. This is actually my feeling. Even in industries, they think academia is waste. They are doing not doing good things, and academia they are also have to gear up, because industries require timelines, and academia should be actually adapted themselves to meet the timelines. Definitely, I mean. Basically, academicians should take some efforts, extra effort to have collaborations. It will definitely help you to grow, if definitely to increase your, of course, funding and all these things. Thank you. If you have any questions, sessions is open now for question answers. I tried to explain as simple as possible for you people. Yeah, thank you.
Okay. Participants, any questions? Or you can type in a chat box.
So no questions means all or none. Either everything is understood or nothing is understood. Yeah, jokes apart. Yeah, somebody is writing now.
Okay, I think so there is no question.
Okay.
Okay, Arundhati, you are there? Okay. On the behalf of the organizing committee, I would like to thank Dr. Sopan, given a nice details on the topic. Thank you so much, sir.
Yeah, thank you for providing opportunity.
Okay, you can
Yeah.
leave any time, sir.
Okay, thank you, sir.
Thank you, sir.
Participants, we have to join at 3 o'clock for a validatory function. And after 10 to 20 minutes, we will conduct a small test. We will send on the WhatsApp group link. You have to submit it back. Okay. Okay, thank you.